Hello, this is a video lecture for microbiology. This was a class that was held on Thursday of last week, February 21st, and it's the first of two 45-minute video lectures. I do apologize, it's taken me so long to get these uploaded and get these done. Ran into some trouble at home, and hopefully you'll be able to view these in a timely fashion. And where we stopped, we're still in chapter 14. Seems like we've been in chapter 14 a while because of the snow day. But anyhow, here we go. We're talking about the second line of defense. And one of the things that hastens the immune response and calls forth immune cells when there is some type of site of infection is chemotaxis. Chemotaxis allows for the proper cells to come in and start to fight against the bad guys, the invading bacteria, viruses, protozoa, whatever, and then also ingestion. Okay, so there are products that the pathogen or parasite is actually secreting that will call for phagocytes. And also the host tissues will start to respond. You'll have neutrophils that the host tissue will respond, the neutrophils will respond by secreting chemoattractants for positive chemotaxis of things like phagocytes. One of the biggest things that is part of the second line of defense, pathogen-associated molecular patterns, or PAMPs, and these serve as red flags for phagocytes and other cells in innate immunity. And I'll show you a slide of how that works in just a minute. These pathogen-associated molecular patterns will slough off of cells, off of bacterial cells. They'll also stay on bacterial cells, but our second line of defense, our innate immunity, has adapted over time to be able to see microbial invaders. Okay? Bacterial PAMPs include peptidoglycan, which you know is the cell wall component, lipopolysaccharide, LPS, which is on gram-negative cell walls, on the outer membrane, and other components. And immune cells have pattern recognition receptors that will actually bind PAMPs directly, and these pattern recognition receptors will then alert more cells in the second line of defense and even recruit cells in the third line of defense in order to bind PAMPs directly. The receptors bind PAMPs, and then they set into motion a defense response that happens with the second line of defense and then goes to the third line of defense. Okay, and I have to move this little alert. Then phagocytes will engulf microbes. They engulf it in a vacuole called a phagosome, and the phagosome itself then will facilitate the digestion of the microbes. Okay, and here's a macrophage, and it has on a toll-like receptor, and it sees a foreign molecule like a PAMP, and it can bind that toll-like receptor, causes the toll-like receptor to close, and that closing process then initiates transcription and translation of cytokines, interleukins, inflammatory mediators. Cytokines, as you know, will promote cell growth, cell differentiation. Interleukins and inflammatory mediators aid in the immune response. Some are also chemotactic. Okay, so this just gives you an idea of how this toll-like receptor will bind a PAMP, and then that will initiate a response from a macrophage to call forth other immune cells and hasten the immune reaction. Now, once a phagosome is formed, it will actually merge with a lysosome. Okay, so lysosomes are the stomach of the cells. They have digestive enzymes, and so they have to be kept in separate vesicles. And in these separate vesicles, then they protect the cells from the digestive components. And then the phagosomes in the phagocyte will fuse with the lysosomes, and then that way the bacteria or the other microbe that's being trapped can be lysed and be digestible. Okay, and in the lysosomes you have granules with antimicrobial chemicals, and they poison the foreign invader, they kill it, and they dismantle the foreign invader. You have to be really careful at this point because capsular bacteria that form capsules, will resist the lysosome, and they can actually hide in the phagocytes and travel around the body. So these granules are effective, but they may not be effective for capsular bacteria. Okay, and this can happen within 30 minutes after contacting the bacteria, and so all of a sudden you're starting to engulf the bad guys and digest them. And so this is definitely a part of innate immunity. It happens very, very quickly. It's the first line of defense, and it doesn't require any type of sensitization of things like T and B cells. Okay, and here is just a representation. This is a picture of a macrophage, and you see lots of lysosomes in there. They're designated with LY, and not all of them have the LY designation, but this is just teeming with lysosomes. And so there's a lot of lysosomic activity in phagocytes, and that allows them to digest at low pH and also other harsh conditions while keeping the cell. Okay, then you have destruction and elimination systems. You have peroxisomes that have monoperoxidase, and these will form halogen ions like chloride ion. Other oxidizing agents, and these are sequestered away from the cell. They're usually in peroxisomes, include hydrogen peroxide, superoxide anion, okay, which is, and other types of free radicals. And these will just oxidize the heck out of the bacteria or the foreign invaders, and this will all happen in the phagolysosome. Okay, and then also liberation of lactic acid, lysozyme, which breaks down peptidoglycan, and nitric oxide, which are antibacterial, and these also inhibit viral replication. And then cation proteins, remember, bacterial membranes are anionic, so if you have a cation protein, that's going to disrupt the membrane. And then finally, exocytosis. So I think I'm repeating some stuff. I'm going to, we talked about fever a little bit, so I'm going to move forward. I think I have covered a lot of this already. So just some of the benefits of fever. This will inhibit the multiplication of temperature-sensitive microbes. These microbes have adapted to your own body. And fever, when the hypothalamus is being reset to a fever state, then that will takes the bacteria out of their favorable range, and that will start to inhibit their multiplication. This reduces the bioavailability of iron. All organisms have iron, and so by reducing the bioavailability of iron, then we're able to kill the organisms. And then by increasing the metabolism, then you're increasing overall immune reactions, not only kill the microbes, but also to suppress them. So there are benefits to fever. You do want to be very careful with high and prolonged fevers. Okay. And when you use something like aspirin or acetaminophen, which is Tylenol, what that does is that lowers the setting of the hypothalamus. So you have to be very careful with doing that because fever, when you have a beneficial fever and you're lowering the fever, then you're actually inhibiting your ability to fight infection. Okay. You can always use a cold bath. Say if a fever becomes too high, if it becomes above 102, then you can start to use these particular techniques because having a prolonged fever and having a very high fever is bad for you. It can cause neurological damage and seizures. Antimicrobial proteins, interferon, and this is a protein that you hear a lot about, therapeutic interferon, and therapeutic interferon is produced using biotechnology, and you have alpha, beta, and gamma interferons, and these are antimicrobial, and our body produces them naturally, so it's part of our body's second line of defense. Okay, this is used for viruses, can be used for other microbes, and it also allows for communication of immune cells. And interferon alpha and beta are produced by many cells, and this will stimulate phagocytosis. And interferon gamma is produced by T cells. Okay, so that's part of the third line of defense. And this is involved in the regulation of macrophages and also T and B cell maturation. Okay, and here is just sort of the way that interferon itself can be, excuse me, the way that interferon can be stimulated. Here you've got a cell that is being invaded by virus. Okay, and this cell is infected. It will probably be sacrificed by cytotoxic T cells. Okay, but upon the infection of viruses, then this stimulates the production of interferon. Interferon is then secreted, and then it will go to neighboring human host cells, and it will allow for the synthesis of antiviral proteins. So it will actually block the virus from being able to invade and take over neighboring host cells. Okay, so the synthesis of antiviral proteins will protect neighboring host cells, will prevent the spread of the virus. Okay, interferon also instructs the development of white blood cells. Interferon alpha activates natural killer cells. Interferon beta helps maturation of T and B cells and inflammation. And then interferon gamma is very useful. It will inhibit certain types of cancer. And so in cancer formation, this is actually going on in your body when you have precancerous cells. Interferon alpha gamma is preventing them from proliferating. This will stimulate B cells, activates macrophages, and enhances the effectiveness of phagocytosis. So interferon gamma has a wide variety of roles. You do not need to know all these different roles, but you should know, that interferon is essential. It's one of the essential small proteins that regulates the immune process. Okay. And then last in this chapter, we're going to talk about complement. And complement gives students fit. It's not necessarily a fun thing for them to work on.